Can everyone hear me? Okay. Well, I want to start by thanking Aman for inviting me out here and organizing this visit and giving a glowing introduction. It's really an honor to be here. Over the years, I've had distant connections with BLI one way or another, as Aman mentioned, and then I've also followed the work very closely. So it's quite an honor to be speaking in such a prestigious institute, and I am really thrilled to be here. As this graphic should illustrate, when I say I'm happy to be here, I really mean it with the utmost sincerity. Boston does have its charm, don't get me wrong, but there's a certain stretch of the year where it's really tough to appreciate that, and I think we're approaching those days right now. So this might be my last chance to actually see the sun for a couple months. So thank you again. So as Aman mentioned, I'm going to talk about some of my work with in vivo microscopy of brain energetics. I've organized my talk such that first I'll talk about some background about cerebral blood flow and metabolism. Then I'll talk a little bit about the materials and methods, the techniques that we use. I imagine, given that this is an optics institute, I won't have to talk in depth about that, but I'll try and get everybody on the same page. Then I'll show some of the results that I've collected, as well as my colleagues, both high-resolution measurements of both intravascular metabolic parameters and also extravascular tissue measurements. So my lab in the Martino Center at MGH, we use a wide range of optical techniques to investigate cerebral energetics at a variety of different length scales and with various degrees of invasiveness, from preclinical investigations in small animals like rats and mice, all the way up to investigating cerebral blood flow in patients in the hospital. I belong to the former half of the group, the one with the microscopy side, so we like to investigate these parameters on a cellular and subcellular level with high resolution in animal models. And this graphic just sort of illustrates the neuronal architecture of the cerebral cortex. It's organized. That constitutes the outer 4 to 5 millimeters of the brain surface, and that's where a lot of higher level brain function, like sensory and perception and motor control, is all regulated. It consists of several different layers, each with different types of neurons, which have different morphologies and serve different purposes. It's very heterogeneous, very dynamic, very energetically demanding, and it requires a consistent, steady, uninterrupted supply of metabolites like glucose and oxygen in order to sustain this very energetically demanding activity. So all these neurons are sort of interspersed with structures like this. There's the overlying, metabolites are supplied by vasculature, which is supplied over the surface of the brain. I'm not sure how well you can see this pointer, but actually maybe I'll just use this. Okay, so the blood vessels start out, they coat the surface, and then they dive into the cortex at various regions, into arterioles, before they branch out into capillary bed, which is very complex and interconnected. And then they drain into venules, which actually ascend and also return on the brain surface. So in order to sustain that, all the very complex neuronal activity, it requires very tightly regulated interactions and signaling between the blood vessels that are there, along with the neurons, obviously, and then glial cells that populate the brain, these astrocytes. So it's important to understand these exactly how these mechanisms operate with great precision to actually get a better handle of other brain imaging techniques that are applicable in humans. And so our weapon of choice is optical microscopy. So we harness the advantages that it's reasonably non-invasive after you've exposed a skull. You get very high spatial and temporal resolution, and you have the capability of monitoring multiple facets of metabolism at once. And all this can be done in vivo under reasonably normal conditions in which the the complex morphological and functional interactions between all these different cell types in the blood vessels remains preserved. So the goal of our work is to actually get a more precise understanding of neurovascular coupling and cell signaling. Because right now it's not well understood, and the end goal is to twofold. Number one, we'd like to use these observations to better interpret observations performed in humans with techniques such as fMRI and near-infrared spectroscopy, which look mainly at downstream hemodynamic responses to stimuli and entail rather convoluted changes in flow, oxygenation, and volume. So the signals that are detected are a little difficult to interpret at this point because there's so many multifaceted and convoluted changes. The other half of it is in order to understand how the pathological breakdown of these mechanisms with the progression of diseases such as neurodegenerative conditions like Alzheimer's and Lou Gehrig's disease and Parkinson's. Also, the aging process involves a lot of degradation of these mechanisms and pathological conditions such as stroke. So this is just a brief simplified overview of cerebral metabolism. The metabolites, glucose and oxygen, those are the primary substrates used to sustain neuronal activity. They're supplied by the blood vessels. And stop here and mention that historically it's been understood that all this transfer takes place at the capillary level. But as I'll show today, we're collecting data that sort of challenges that conventional notion. But the breakdown takes place within the cytosol with glycolysis that yields a couple of molecules of adenosine triphosphate, ATP, which is the primary substrate for cellular work. And then the majority of it gets broken down in the mitochondria, first through the Krebs cycle, in which the electrons are stripped from the precursor molecules from glucose, and those electrons are then transported to the electron transport chain and used to drive ATP synthesis through the oxidation or through the formation of water by donating electrons to oxygen. I imagine some of this is well understood, but couldn't hurt to go over it initially. So the point is that with the progression of several diseases, there's several stages, several steps of these get compromised, and in order to better understand the etiology of these diseases, we'd like to be able to pinpoint exactly where these take place. And we'd like, hopefully, optical microscopy can help resolve some of these. Not only that, but ultimately these observations should help identify new biomarkers, because potentially, reportedly, some of these metabolic changes actually precede the clinically observable symptoms of conditions like Alzheimer's disease. So it could lead to new diagnostic tests and also possibly potentially new preventative therapeutic techniques and strategies. So that's the overarching goal of our work. Now on to some of the techniques that we use to actually investigate this. My primary tool of choice is laser scanning microscopy. I imagine most of you are very well versed in the benefits of confocal and two-photon imaging. This is a very somewhat generic illustration now of showing that two-photon excitation, the primary benefit of it involves getting enhanced spatial resolution coming from the fact that nonlinear excitation causes a very small volume of your sample to get excited. So there's signal from outside of the focal spot is eliminated. In confocal imaging, which we also utilize, we can get similar spatial sectioning, but it requires spatial filtering of the detected signal. There are advantages and disadvantages to techniques. Confocal is less expensive, and it's applicable to a wider range of reagents. But two-photon is basically the way of the future, and it's very widely utilized now. So I imagine I don't have to go into too much more detail about it. So we couple laser scanning microscopy with lifetime-based measurements. Rather than looking at just the intensity, we look at the time-resolved decay of either fluorescence or phosphorescence. And after modeling it as the sum of either one or multiple exponential decays. We can use the lifetime information, this computed value tau, to give us a more comprehensive information about the microenvironmental conditions of the fluorophore. It gives you variations in these parameters will tell you information about the fluor, including changes in temperature or viscosity, and we use it primarily for looking at changes in ambient oxygen tension, and for and I'm developing methods to make it applicable to looking at enzymatic binding of intrinsic fluorophores, as I'll go into later. So it provides, in principle, a more comprehensive information than standard intensity-based imaging, but it does require more sophisticated technology and analysis techniques, including nonlinear fitting to a multi-exponential decay and deconvolving with instrument response function. So that is the general technique. We use it coupled with the application of certain oxygen-sensitive phosphors. This molecule is oxy-4-R2, developed by our collaborator Sergei Vinogradov, who's a brilliant biophysicist at University of Pennsylvania. Basically, the oxygen-sensitive platinum porphyrin or metalloporphyrin core phosphoresces, and as illustrated in this Jablonski diagram, if there's ambient oxygen available, that will scavenge, or it will quench the phosphorescence, steal the excitation from the excited porphyrin molecule. So as a result, there's less phosphorescence emitted in the presence of higher oxygen, as illustrated by this figure. This is the phosphorescence decay measured of oxy-4-R2 in an artery with high oxygenation, and this is the same. This is from a vein nearby in the same animal, separated by a few hundred microns, I think, at the surface. But there's a greater amount of phosphorescence in lower oxygen conditions, and so consequently the lifetime is longer. Using this information and an empirical relationship, the Stern-Volmer relationship, you get quantitative information about the actual how much ambient oxygen is available in the microenvironment around the phosphor. So I've used this quite extensively, this molecule, with confocal microscopy to measure intravascular oxygenation. But more recently, slightly more recently, my collaborator Sava has used a more beefed up version of more beefed up oxygen-sensitive probe, also developed by Sergei, that extends its utility to 2-photon imaging. Basically, the probe is called PTPC343. It has the same oxygen-sensitive platinum porphyrin core, I believe, or something similar. But since these things cannot be excited easily by 2-photon excitation, it's clustered together with some 2-photon excitable coumarin antenna molecules, which absorb 2-photon excitation and transfer their excitation to the phosphor via FRET. On top of that, Sergei spent an extensive amount of time developing a dendrimeric cloud around these the oxygen-sensitive core to make it more extend its utility to be able to actually measure in extravascular space, which was quite a breakthrough for brain energetics. But also it reduces its sensitivity to other factors that may otherwise affect its lifetime, such as pH and temperature and things like that. So the mechanism is essentially similar, except first there's a FRET of the coumarin molecule, then there's phosphorescence from the PTP, but that will get quenched by the ambient oxygen. And these are some of the spectral properties of all the fluorescent and phosphorescent molecules within the probe. So together with that, we also utilize optical coherence tomography. I imagine many of you are familiar with that. In a nutshell, it's very similar to ultrasound. It relies on the back-reflected light from the sample. It gets back-reflected coherent light, which gets recombined with the sample arm, and the interference patterns will give you information on the scattering properties of your tissue. And if you look at the Doppler shift in the signal, then you can use it to get information about the velocity of scattering particles. And if you, my colleague Vivek, who's now at UC Davis, used this, extended this to be able to measure actual absolute microvascular blood flow as opposed to just relative velocities by integrating the velocity over the cross-sectional area of a blood vessel. So, between measuring PO2 and measuring blood flow, those served as the primary motivations for us developing our multimodal optical imaging system, coupling OCT together with laser scanning microscopy. This was my first task as a postdoc to help design and construct this, and this is the original configuration of what I now call the octopus. It's capable of, simultaneously capable of measuring PO2 with confocal imaging and microvascular blood flow with OCT, and separately it can be used for two-photon imaging and also of oxygenation, but at deeper locations in the brain and also in extravascular space. And it can also, and then later on you can use OCT to give you microvascular blood flow and morphological information about the vasculature. So this system has been basically a workhorse for me for the past several years, and it's sort of constantly in flux. Since its original construction, we've modified it a bit to extending its utility and incorporated other modalities into it. So this is a more modular figure and updated. Still has the same three original capabilities, but now we've actually added the ability to do laser speckle imaging and optical intrinsic signal imaging. This just illustrates how the two separate scan paths for OCT and laser scanning microscopy are coupled together with a very custom-made dichroic, and how the fluorescence intensity from two-photon imaging is separated early on to optimize the amount of collected signal, as well as using wide F-and-do optics to collect as much as possible, enabling us to image deeper within tissue. I've also recently added the capability of being able to simultaneously measure EEG and also coupling this with TC-SPC equipment commercially available from various film companies. So it's essentially constantly in flux. And I've used it quite extensively for all the studies that I'll describe here. So in addition to those techniques, our experiments require some very precise and careful surgical preparations in animals. This is a cranial window, a craniotomy done on a rat. We do it both in rats and mice. We try and control as many physiological signals as possible in most cases, so it requires a tracheotomy and monitoring blood gas levels through cannulation of the femoral artery and vein. Generally, our animals are anesthetized with either isoflurane or anesthesia, and we monitor these signals such as arterial blood pressure and body temperature and expired CO2 continuously throughout the preparation and the actual experiment. It's important to do this because in order to do extensive physiological studies, you need to have as tightly regulated and as robust preparations as possible. So this is all a very non-trivial task, and I have a team of technicians to thank for the data that I've collected and I'll present here. So to do fluorescence measurements or phosphorescence measurements, it requires either an intravascular injection of PO2 sensitive dyes or a dye that will give you some indication of change in metabolism. So we've also injected into the tissue using a picospritzer pump materials that are sensitive to calcium signaling and also mitochondrial membrane potential, but I won't go into too much detail here. Yeah, so those are that's an overview of our materials and methods. Now I'll just go into some observations. Using the octopus, I was able to do a nice study of simultaneous PO2 and blood flow over the entire cranial window. Basically, we had the ability to monitor in individual microvessels what the actual oxygenation levels were, and at the same locations measured the absolute blood flow, and we were hoping that would give us some indication, or from that we were hoping to be able to calculate parameters such as the cerebral metabolic rate of oxygen. That last part didn't work out so well, but still it was a very elegant study that allowed us to measure things with high spatial and temporal resolution. So some things worth mentioning here. Like I said before, these signals up here are all from phosphorescence intensity of our intravascular dye, whereas these angiograms come from the Doppler signal of the intrinsic scatterers, the magnitude of them. So in this case, because of the higher oxygenation in arterioles, they appear brighter in the confocal images, whereas because they move faster, with the scattering signal is higher, so their arterioles are brighter down here. Also of interest is the fact that prior to these microscopic measurements that we had performed, the gold standard of measuring oxygen tension in tissue was using a Clark electrode, which was very invasive and noisy as well. And people historically have had lots of difficulty measuring multiple locations in a given sample because they have to take a measurement and then very carefully reposition it, and essentially they poke a lot of holes in the process, so it's not unclear how the viability of the tissue is sustained during all those manipulations. In this case, it's not necessary to do that. It's less invasive, but nevertheless, we still managed to observe considerable heterogeneity even within individual vessels and over the course of several hundred microns, which replicates the observations that they saw in using Clark electrodes. And at that point, they had assumed it had to do with just how invasive the procedure was, but there's actually considerable heterogeneity in PO2 within a very small distance of the brain surface. And this is also important because this whole field of view would constitute maybe 1 or 2 voxels in more non-invasive modalities such as PET or fMRI, which measures similar information about oxygenation. So what they're measuring with those techniques is typically a very spatially average value of a signal that varies quite considerably over a much smaller distance. Yes? A higher velocity. Yeah. So this is the amplitude of the scatter, I think, or the amplitude of the frequency shift. How does that work? To be honest, I haven't. It's been a while since I've looked into this, but I'd have to look back at the algorithms and get back to you. But I believe these are just. This isn't like raw Doppler signal here. It's been processed and converted to velocities. It's right, the subsequent calculation, right. Sorry, I should have clarified that. Right, so this illustrates a couple of interesting points. First, the relationship between vessel diameter and blood flow follows a parabolic routine. That was the best fit that we got, and that's somewhat satisfactory, I think. Originally we thought it was linear, and it turns out to be parabolic, parabolic relationship. But to me, more interestingly, is a subject which I'll return to many times today, the observation that smaller diameter arterioles have lower PO2 values, which strongly suggests that conventional notion of all oxygen being delivered by capillaries is not entirely accurate. This is consistent with other recent studies, and it very strongly suggests that arterioles play a more pronounced role in oxygen delivery than originally imagined and originally assumed. We also noticed there was a slight trend in increasing PO2 with increasing venules, which suggests it mildly suggests the presence of some sort of oxygen efflux from the tissue back into the veins. And we've, my colleagues Sava and Anna DeVore, they've both observe this periodically. It's not something that's consistent, but it does happen sometimes, a certain percentage of the time. So that's also a very interesting observation. Yes?
Does that have anything to do with the anesthesia that's on board?
Potentially, but I haven't tested different types of anesthesia, so I couldn't say for sure whether or not it's... this was all under alpha-chlorylose anesthesia, which is not... I believe its influence on oxygenation I don't think has been characterized very well. But that is a good question. Isoflurane, however, that's a vasodilator, so these relationships may not be as applicable because the increased flow will lead to decreased oxygen extraction. Okay, so those are the observations that we collected under baseline conditions. Adding on to that, my PI David was always somewhat intrigued by this observation that if you quantify the amount of oxygen extracted and correlate that with the amount of flow in ascending venules, it's a very linear relationship, suggesting that there's a larger amount of drainage from how to put this? Well, they're very well matched, the level of drainage and the level of oxygen extraction. So it has some sort of implications about angiogenesis and it's very consistent with observations that other people in our lab have collected with changes in SO2 with increased angiogenesis from brain development as infants progress. So it was just noteworthy to him. Doesn't really resonate with me as well, so I don't really emphasize that as much. So for the same study, we were able to, those previous measurements that I showed, they were sort of averaged over the course of 2 minutes. However, you can increase the temporal resolution by just measuring a B scan in OCT, a single line, and then measuring the PO2 at the corresponding ascending and descending venules and arterioles in that plane of view. And we were able to harness this to measure how oxygenation and blood flow change in response to somatosensory stimulation. It's more pronounced in venules generally, which is unsurprising, but there is some small response in arterioles. These first 2 points show that there is a subtle increase in oxygenation, which sort of reinforces the idea that there's with an increase in flow comes you're flooding out some of the deoxyhemoglobin, and so that means that previously there was some oxygen being extracted. It sort of leads to a shift in how different types of microvessels contribute to oxygen delivery under stimulated conditions. So it was an interesting observation, but also we were able to quantify delays between the increase in flow and the increase in oxygenation. That gave us some. Oh, yeah, that's this slide. Here are some of the delays that we observed. In arterioles, there's a very subtle delay between the two, but there's also a delay between PO2 changes of arterioles and venules, and that will give you information about the general transit time through the vascular bed in response to functional stimulation. But also these subtle delays between flow and PO2 will sort of reinforce that. Yeah, I'm not sure if there's much more to ascertain from this, but the onset times were also quite interesting to. Well, those give you more information about transit time, I suppose. Right. So those are observations that I collected a few years ago and were very interesting. But my colleague Sava recently used the 2-photon phosphor and did a very comprehensive and elegant study where he measured PO2 throughout the vascular network up to a depth of about 300 or 400 microns, tracing it from the peel surface down into the capillary bed. And using this and OCT for measuring microvascular blood flow and extensive modeling about the vascular network, he was able to make some very interesting observations about oxygen delivery and how different vessel segments within the vascular bed contribute to that. So again, this is a cross-section of a diving arteriole. If you measure, you'll notice that the oxygen tension is higher within the middle of it compared to the boundary, again reinforcing that idea. But I think I have a better image later which shows an interesting observation that Salva collected and is consistent with some of our colleagues in the field, like Karl Kashish, where if you look at a diving arteriole in the cortex, there's more often than not, there's a cylinder of tissue around it where there's no other vessels contributing to it, and no vessels nearby, which suggests that actually all of that tissue within that cylinder around the arteriole is being oxygenated by the diving arteriole. I'll return to that in a moment. So, as I said, Salva collected a very extensive amount of data about the PO2 in different vessel segments, and then converted that to SO2. And based on his modeling work, it suggests that actually, contrary to what was the conventional notion that capillaries deliver all the oxygen, it turns out under baseline conditions, almost 50% of the oxygen is delivered by arterioles. So this graphic sort of illustrates that as the arteriole dives and branches out, the yellow region corresponds to the territories that are being oxygenated by different vessels, and the color of the vessel corresponds to the actual amount of SO2, the amount of the concentration of oxygenated hemoglobin compared to total hemoglobin. And under baseline conditions, most of the supply actually comes in the earlier segments. And as he showed with hypercapnia experiments where the flow is increased, it turns out there's some sort of dynamic shift involved when there's an increase in blood flow, and it's only under stimulated conditions or metabolic perturbations that these downstream segments actually contribute substantially to oxygen delivery. So this was just recently accepted, like last week, I think. So more information should be available about that soon. So in summary about the confocal the intravascular measurements, we have the capability of measuring blood flow and oxygenation simultaneously in individual microvessels. The values demonstrate a lot of heterogeneity in a very small volume. There are some implications about the regulation between oxygen extraction and the vessel diameter and possible angiogenesis, but that needs to be explored more thoroughly, I think. And yet again, because there's a contribution of oxygen delivery from arterioles, this has implications about people's understanding of the hemodynamic response and how to interpret the bold signal fMRI. Typically, some sort of stimulus will induce very drastic changes in cerebral blood flow and cerebral metabolic rate of oxygen. Generally, it's been assumed that a lot of the observations that have been collected with fMRI are largely come from, or almost primarily from contributions from the veins and the small venules, but this data suggests that that may not be true. The arterioles may contribute in the earlier part of the temporal response, and they may play a much more profound role than originally believed. So this is shedding new light on it and on the concept of the hemodynamic response and the role that different vessel segments play within oxygen metabolism and metabolite supply in general. It would be phenomenal if we could couple this with measurements of glucose delivery, and there are some fluorescent glucose dyes, but they don't necessarily reflect glucose consumption as well as one would hope. They sort of get taken up by cells and get trapped there, but they don't get further broken down. So interpreting the signal could be kind of challenging. But moving on to some extravascular measurements, which have been performed more recently. again, I'll show some observations from my colleagues Sava and Anna about the PO2, and then I'll talk more about intrinsic autofluorescence in depth, I guess. So as I said before, this is a better illustration of the concept that I mentioned about arterioles being the sole supply of oxygen for certain cylinders of tissue within the cortical surface. If you inject the PTPC343 in the brain tissue and then measure in extravascular regions around these vessels, you'll notice like pretty drastic gradients in oxygenation in surrounding these arterioles, which yet again reinforces what I said before. But in addition to that, it actually, since there are no other sources of oxygen in the area, it opens up the possibility to be able to measure this parameter, cerebral metabolic rate of oxygen, using one sole parameter, PO2. That's quite profound because previously with these other techniques you need some measurements of oxygenation and blood flow, and you need to account for a couple of confounds in certain ways. So this has quite a bit of potential of being able to measure CMR02 more easily and more accurately and with higher spatial resolution. So I believe he's conducting work like that right now, doing more extensive work on the influence of different metabolic perturbations on CMR02. There's no telling how homogeneous or heterogeneous CMR02 is, so it remains to be seen exactly how useful it is, but nevertheless it is opening some doors. Again, here's more evidence suggesting oxygen efflux from the veins. The fact that there's higher oxygen, actually this is suggesting that there's oxygen delivery from veins, but the gradient is a lot more subtle and it's not always observed, so this needs to be characterized more extensively, I suppose, but it's a perplexing result. So my colleague Anna used the PTPC343 to measure changes in oxygenation during a somatosensory stimulation. Again, she stimulated the forepot for either 2 seconds or 20 seconds and measured the traces that look very similar to the hemodynamic response, but they're actually actual values of the level of PO2 in the tissue. And what was interesting in this study is the fact that at certain locations, again, the baseline PO2 is heterogeneous and it varies with proximity to oxygen supplying vasculature, but at regions that are somewhat distant from this oxygen supply, which have a low baseline PO2, the overall amplitude of the response is somewhat negligible compared to better oxygenated regions. Now, she analyzed this a little more and sort of suggested the idea that conventionally challenging the conventional understanding of the hemodynamic response. Typically, a localized stimulus to the brain or to the cortex will lead to an increase in cerebral blood flow that's disproportionately larger than the cerebral metabolic rate of oxygen. That was an observation that was originally collected using PET in the mid-'80s. So the idea is that a surplus of oxygen is delivered to the tissue. But with these measurements, which were the first of their kind to be measured on such a high length scale, the idea emerged that actual regions that are distant from oxygen supply, since their increase in oxygenation is so marginal, it suggests that what we initially thought was overabundance of oxygen, where they were flooding the brain tissue with oxygen, and maybe just barely what's necessary to prevent more distant regions from becoming hypoxic. So that it's food for thought at this point. It needs to be characterized more clearly, but it is nevertheless intriguing, and it's prior to this observation, people have had other theories about what purpose this surplus of oxygen would serve, but
so just to make sure I understand, those are distances from the 0 to 8, 8 to 12.
These are the grouped average baseline PO2s, actually. These plots, there's no actual distance from the oxygen vessels, but the ones that have lower baseline values are typically further away from oxygen supplying vessels. She also observed a difference between a 2-second long stimulus and a 20-second long stimulus. That's 2 second is the solid lines. And that has implications about how CMR2 might evolve over the course of a long stimulus, something that also needs to be characterized a little further. But nevertheless, this study was somewhat the first of many that should come to characterize this further. It's worth pointing out now, this is the last slide I'll show with, I think, with this PTP C343 dye. All these observations are very eye-opening and very compelling to the neuroscience community, so everybody wants to do them, but unfortunately it suffers from the disadvantage of very limited availability of this dye. Sergey was able to make quite a bit of it several years ago, but since then the resources haven't come through to produce them in the volumes that people would want, and also we have a limited supply that we've been sort of used very sparingly for the past few years. Remains to be seen how these studies will evolve over time, but that's something, even though the synthesis process has been published, no one has been able to replicate it well, I don't think, or no one has published any studies yet where they've used the same process, but hopefully in the future that all of that will change. Okay, moving on to some more what I've devoted the past few years to. I sort of switched off from looking at intravascular measurements of blood flow and oxygenation, and I wanted to look at more direct markers of energy metabolism in the tissues. So like many people here, I think, we started looking at how the autofluorescence of the intrinsic electron carrier NADH. So nicotinamide adenine dinucleotide. I imagine this is a fairly mature technology, I think, or at least observing the intensity of the fluorescence has been around for close to 60 years, I believe. It was developed and formulated by Britt and Chance back in the 50s and 60s. Well, NADH, it serves several roles, actually, in glucose breakdown, ATP synthesis. It's involved in glycolysis. It gets formed here. It gets formed in the Krebs cycle, and then it gets oxidized, stripped down back to NAD in the electron transport chain. So it plays multiple roles in the process. So observing its intensity, interpreting that, and correlating it back to different steps of oxidative metabolism is a bit unclear, I think. And yeah, I guess this slide just sort of reiterates that. The signal that you get reportedly comes primarily from the mitochondria, but a direct correlation between the fluorescence intensity and the change in the relative shifts in different steps of ATP synthesis remains unclear right now. Nevertheless, it's been used quite a bit, first as single photon based for several years, and then over the past few years it's been used in both in vivo and in a whole bunch of other slices and cell cultures using 2-photon microscopy, the intensity based. And more recently there's a lot of work, lifetime-based work too. This is an illustration, this is a fluorescence image with the vasculature labeled color-coded, collected by my colleague Karl Kosciuszko. He and several other people have shown that you'll get bright patches of NADH, which corresponds or which can be correlated with a deficit in oxygen or an increase in metabolic demand. And you'll see this in regions far away from blood vessels again. And in the case of my colleague Hannah in UCSD, she saw relatively bright patches around diving venules, which, to be honest, all of this has been collected in vivo in mice, and I work primarily in rats, and I've spent years trying to see something similar to this, and I've never been able to see such distinct patches like this. Initially, it used to frustrate me, but now it kind of suggests that if there's more uniformity in the images that I'm seeing, then there's less likelihood of metabolic deficits or hypoxia. So perhaps the rats are a bit more rugged and stable. So I'm a bit more comfortable with the fact that I don't see these patches anymore. But also, I've decided, I've devoted a lot of time to looking into NADH lifetime measurements rather than intensity-based. And the purpose behind that, there's a reasonably large body of literature on it now, where if you look at the fluorescence decay of NADH, you can decompose it into the sum of several different components, and each of those reportedly corresponds to different enzymatic formulations of NADH, some of which may give you a better indication or may be better correlated with different steps of either glycolysis or oxidative metabolism. So while all the people who look at NADH phlegm agree that it can be decomposed or it can be resolved into multiple components, the actual number of components that they see has been the subject of dispute. Well, this just serves to show that in order to do these measurements, I modified the octopus such that it could, all my signals went to commercial TCSPC, time-correlated single photon counting boards rather than what I previously used. And so there's commercial software that I use to obtain all this data. Anyways, back to the number of components. I believe there are people here who, and other parts of the country who typically report the NADH phlegm is the sum of two components, so it's a bi-exponential decay. And they like to attribute the shorter lifetime component to a formulation of NADH that's not bound to any enzyme, and the one longer component that they see, they just call that protein-bound NADH, or enzyme-bound NADH. All the work that I've done, whenever I collect these histograms, I always see there's always distinctly four components. And this is consistent with one other person who's explores this in brain tissue. So that's how I investigated. Also, there have been some chemistry reports where they looked at just NADH in solution, and even they suggest that NADH on its own, in the absence of any proteins or enzymes, even that has two distinct lifetimes because of presumably because of the molecular conformation of NADH. It tends to quench itself if it's folded up in a certain way. So even NADH alone can be modeled with a bi-exponential decay. Yes?
Does that have to do with is it extra mitochondrial NADH versus inside the mitochondria? So is it possibly that NADH is involved in lactate metabolism?
So could you be more, what do you mean this? Like the presence of different components or the?
The two lifetimes that you're with free NADH. Does that have anything to do with the location?
That's what's been largely asserted, and that's something that I would love to get more definitive clarification on. And people, like I said, people have implied it, but it's very spurious, the observations that they made. So it kind of depends on who you ask. I need more convincing. More importantly, going back to the bound versus unbound, I have some collaborators at MGH who are they study mitochondrial physiology and mitochondrial genomics, and when I mentioned to them the idea of just bound versus protein bound, they kind of laughed in my face because there was to a biologist that's so nonspecific, so vague that it really doesn't tell them anything. There's actually hundreds of enzymes within a mitochondria that NADH can bind to, so just having a perfectly binary idea like that does not sit well with biologists, basically. And I don't think 4 is any more reassuring to them, but that's consistently what I've seen, and that's how that's my understanding, I think. It bothers me that there's not a better consensus about this, but it really could depend on what tissue you're looking at and how metabolically active they are, and also the actual instrument itself. Lifetime fitting is it's not as straightforward as it should be, I think. But I'll stop there before yeah, we'll just stop there. Anyway, so I observed 4 components, and more importantly, one thing that sort of reassured me about it, I've seen it in brain slices and in vivo, and when I expose both brain slices and or a live animal to some sort of metabolic perturbation, like exposing the brain slices to some sort of metabolic inhibitor or just depleting the oxygen of the animal, there's like varying level of sensitivities of these 4 components. Some of them seem to diminish, some of them seem to go up. I'm reasonably convinced that there's at least more than 2 components. I've had good results fitting with 3 and 4, but it remains to be seen exactly which one's the correct one. And I would actually caution someone from fitting a multi-exponential decay with more than 3 or 4 components, because it becomes a bit very uncertain, to say the least. A nonlinear fit, you need a lot more photons to be able to resolve that many components. This is more evidence of the differential sensitivity. This was collected in vivo. These are the NADH intensity images that I collect, it's the integrated intensity from the time-resolved decays at every single one of these pixels. This is a corresponding image of supposedly metabolically inert SR101 sulfurrhodamine dye, which gives me information about the morphology of the presence of astrocytes or neuropil or blood vessels within the region that I'm interrogating. So that doesn't change over time, but the relative amounts of different components of NADH change, and the brightness of NADH changes as I modulate the amount of oxygen that's inhaled by the animal. So with the understanding that there's 4 components in the brain, consistent with the previous reports on NADH film in the brain, I had to develop my own processing software, because the commercial one only goes up to 3. So I've spent a lot of time developing these analytical techniques, and also I'm interested in trying to measure how the relative amounts of these constituents and how they evolve over time in response to different metabolic stimuli, you could say. So as a consequence, I did a study where I was measuring the evolution of the 4 different NADH components, actually 5, because if you count the offset incorporated into the model, how they changed over time in response to anoxia, a 45-minute breath hold period in a rat, and did some nice analysis that showed that they vary, the various parameters of how these profiles change, like the onset time and the total change and the rise time, some of them differ significantly from others in response to oxygen depletion. But in order to collect data with this type of temporal resolution, I had to trade off the spatial resolution. So I was imaging an entire field of view with just a 32 by 32 pixel resolution. And that allowed me to image every 2.5 seconds. And then I had to integrate the photons from the entire field of view to perform these fits. I went on later to try and, I was hoping to get, like I did for the PO2 measurements, be able to measure in distinct points. So again, trade off spatial resolution for better temporal resolution. So I had to work out a process where I was able to trigger the commercial software independently and measure in these patterns similar to what we do for PO2. And to be honest, unlike those observations that previous people have shown with intensity, I have not seen consistent gradients in NADH around the vessels. I mean, there are definite gradients, but in some cases you get an increase in NADH away from arteries, and in some cases you'll get a decrease. And the same happens to be true with veins. So I think a combination between the very low signal to noise that I get and the somewhat uncertainty of the fit, I wasn't comfortable pursuing this much more, I think. Instead, I think it's a better utilization of my time to try and figure out to actually validate the significance of the distinct components that I saw. I spent the last few months working on a study where I tried to modulate metabolism in vivo in the rat brain with the administration of classical reagents that are used to change metabolism, used in mitochondrial studies from the past. So I had to devise a way I changed the cranial window chambers such that there was an inlet and outlet ports for liquid reagents, and then using a gravity feed, we were able to deliver the reagent to the brain surface directly. And this has my initial goal was, or my initial attempts, I had just injected them intraperitoneally or intravascularly, but that has such far-reaching global effects on the animal's well-being, and this is a lot more localized. So actually getting this working properly took a reasonable amount of time. Nevertheless, at some point it became more stable and we were robust, so we used it to test the influence of the 4 different components in the presence of a whole bunch of different reagents. So in the first case, just as a control, we just delivered artificial cerebral spinal fluid. That's it's similar to what the neurons are bathed in the cerebral spinal fluid. It's the same salt concentration. It's not supposed to have any metabolic effect, and there were no changes in the overall amounts of the different components or the fractional fluorescence, which gives me some sort of indication of the relative amounts of each component with respect to each other. And unfortunately, no change in overall intensity either. Next, I tried to block glycolysis using 2-deoxyglucose and iodoacetic acid. And again, there was the changes were somewhat subtle, but in certain components there were some changes, including a reduction in this third component. Not really sure why one caused an increase and one caused essentially no increase or no change in intensity, but something I need to investigate further. Next, I tried to use sodium fluoroacetate to inhibit NADH production in the mitochondria, where NADH is produced more readily, and that led to a much more drastic change, a decrease in NADH, unsurprisingly, and a change, not a substantial change, but it was different across the different components, so the relative amounts changed. I guess the most pronounced one would be in level component 1 and 2. I should mention I'm still processing this data, and hopefully it'll tell a more comprehensive story when I'm done analyzing it. I also blocked the electron transport chain using either cyanide or rotenone. So this prevents oxygen consumption in the case of cyanide, or more upstream prevents NADH from being oxidized back to NAD at complex 1 in the electron transport chain. And that led to drastic increases in NADH, unsurprisingly, because the bottleneck is right here, right after NADH is synthesized. And again, there was variability within the relative changes of the different components and how they contribute to the overall fluorescence signal. And lastly, I tried an uncoupler between the electron transport chain and the actual final step of ATP synthesis, phosphorylation. And that led to a similar effect as blocking NADH over in the TCA cycle, at least in terms of intensity. It might be seems a little different in terms of the relative overall amounts of fractional fluorescence. So looking at them all together, it seems like I'm finally able to confidently say that NADH phlegm actually does have some ability to resolve different variations in mitochondrial function and potential bottlenecks at different stages of glucose breakdown. Happy with this, but it needs to be investigated further, and I'm giving thought to looking at it, just analyzing it in terms of 3 components as well, and seeing if the same trends exist. And also, as a sort of a demonstration to couple along with this, I've started to, instead of delivering classical mitochondrial inhibitors, I'm looking into ways to induce localized focal seizures as models of epileptic seizures and see how that metabolic perturbation, whether I can relate that back to any one of these changes and see exactly what happens within the mitochondria during a seizure. Collected some data on that yet, but so far I need to collect more for statistical significance. I believe that's it. So yeah, in summary, the PO2 spatial variation, this was very novel, turned a lot of heads in the neuroscience community being able to measure extravascular PO2, and it's opening doors to help interpret and gather information about CMR02 on a much finer length scales. Anna's data about functional hyperemia it's possibly answering some questions and creating other questions about the actual purpose behind the uncoupling between oxygen metabolism and blood flow induced by a localized functional stimulus. My NADH work, I don't see these spatial gradients in terms of the different NADH components as a function of proximity to vasculature. It's not as pronounced as what other people have reported, but this new study shows that it is quite useful for trying to better resolve or better pinpoint metabolic changes with more precision in terms of its metabolic significance. So that hopefully should give us a more comprehensive information about metabolic behavior as opposed to just whether there's an oxygen deficit or not, which is primarily how people interpret NADH intensity. Looking forward, hope to improve upon these studies by developing ways to implement 3-photon microscopy. That'll give us the ability to measure deeper within the cortex and give us more information. That's where the neuroscientists really want to get information. That's where the magic happens, in the deeper layer of the cortex. Also want to couple this technology with with transgenic models representing different diseases of humans, like Alzheimer's and Parkinson's. That should give us more information about how those diseases tend to progress. Also, we want to start incorporating new paradigms so that we don't have to anesthetize the animal, and possibly we can perform more longitudinal studies to see how these diseases progress over time. And as time progresses, we'll also probably couple more modes to get a more comprehensive, thorough picture of the metabolic activity going on. So a lot of this has been a team effort. There's a lot of technicians, Bouillon and Wei and Lana. They've helped out enormously with preparing the animals. Sava and Jungwon have helped quite a bit collecting the data, interpreting it, and giving a lot of good advice, as is Anna and Hannah. They're my collaborators at UCSD, and they have done a lot of work in NADH and PO2 in mice and rats in their lab. And Vivek and Harsha, they're formerly members of our lab, but now they're out at UC Davis, and they've been instrumental with all the OCT aspects of this work. So I guess with that, I'll conclude. This actually, as Aman mentioned before, I do actually have a pretty good reputation for these kinds of things. This used to be my concluding slide, but I figured I'd update it. It's been a while. This was back in 2011. And this one's more appropriate, I think, because it features BLI alumni. I don't know if you guys can recognize. That's Darren and that's Aaron. This was actually just last Friday, and this was from a year ago. So with that, I'll take any questions and comments.